Today's video, video 8 in the DNA unit, is going to be all about mutations. So, a mutation, by definition, is any change in the nucleotide sequence of DNA. So, in other words, if you change an A to a T in your DNA sequence, that would be an example of a mutation. And mutations can be caused by many things. Remember we said DNA polymerase makes an error during DNA replication about 1 in every 4 billion times. So that would be a mutation. Things like carcinogens, so things like UV light, certain chemicals that are found in cigarettes, these things can also literally change your sequence in your DNA. They can literally change one base to another. So the size of mutations vary wildly. They can be anywhere from changing one base to changing an entire region of a chromosome. And mutations can also be the cause of many disorders and diseases such as sickle cell anemia. So mutations are changes in your DNA, and we know that we already talked about in a previous unit how they could cause things like cancer if the mutations are in a gene that's important for cell cycle control. And we know that mutations can cause other diseases as well, but the question is, are they always bad? So I'll tell you, probably about 99% of the times, mutations do nothing. They're neutral. And then, kind of second most likely, they're harmful. But every once in a while, you get a mutation that's actually beneficial. And mutations are also fairly common. About 1 in every 100 egg or sperms carries a mutation. So like I mentioned before, every once in a while, you can get a mutation that's actually helpful or beneficial, and it can help to fuel evolution. So, because remember, mutations are really the ultimate source of variation in a population. So maybe I had a mutation in a gene that was responsible for beak shape in a bird, for example. If I got a mutation that gave me a slightly different shape in the beak, and that beak shape made it easier for birds to eat seeds, that might be a beneficial mutation. So sometimes mutations are helpful, and they may spread through a population as parents give those genes to their offspring, and so on and so on. So we know that DNA directly codes for the sequence of amino acids in a protein, and we know that amino acids dictate the way a protein folds, and we also know that the shape of a protein dictates its function. So therefore, mutations are problematic because if you have a mutation in the DNA, you're going to have a differently shaped protein and therefore a different phenotype. So ultimately, the problem with mutations is that they can create non-functioning or malfunctioning proteins. So there are many different types of mutations, and the first type we're going to look at is small scale, or mutations that are altering a single base, or we're kind of looking at the base level. So the first type is either called a point mutation or a base pair substitution. And this is basically when you take one nucleotide and you substitute it for another one. So maybe I have a DNA sequence where I'm supposed to have an A, but instead I have a G substituted in for that A. And a second type of mutation we're going to look at is what's called a Frameshift mutation. So this is not where you change one base to another, but instead it's where you add an extra nucleotide, so that's called an insertion, or you delete an entire nucleotide, and that's called a Frameshift deletion. And this does something called shift the reading frame, which we'll look at a little bit on the next slide. So between these two, one is significantly more serious than the other, and we'll see on the next slide which one that is. So up here, I have a normal situation without any mutation. So here's my normal DNA sequence, and I can transcribe it into RNA, and then I can read the RNA one codon at a time and make my protein. So the first mutation type that we looked at on the previous slide was a point mutation. So I'm going to change just one base. I'm going to change this C here to a G. And if you look, when I go to make my RNA, whereas before I had a G here, now I'm going to have a C in my RNA. And then if you look at my protein originally was methionine, alanine, tyrosine, proline, and then tyrosine again. But now my alanine, because I have a different set of codons, now is a proline. So in this point mutation, I've changed just one amino acid. So contrastingly, if you look at a Frameshift mutation, here, instead of changing one base to another, I'm inserting an extra C. I'm putting an extra letter into my DNA sequence. So now, I also have an extra letter in my RNA. So what's different about a Frameshift mutation is if you compare the codons in this RNA to the codons in my original RNA, I'm reading completely different codons. Because I've inserted one base, everything after this base has been shifted over one. So now every single amino acid after that point has been altered. So I call this shifting my reading frame. I'm reading in different sets of 3 by inserting or deleting a base. So to go back and answer our question, the more severe mutation type would be a Frameshift because it affects every single amino acid after the mutation. In this case, we had an insertion. And if we look at this question, what would be the least damaging Frameshift mutation if you inserted 1, 2, or 3 bases? And the answer to that, if you inserted 3 bases, you would still be in the same reading frame. You would just have an extra amino acid. So that would be the least damaging of the 3 types. So we can actually further classify point mutations. So there are 3 types of point mutations. There are what we call nonsense mutations, missense mutations, and silent mutations. So let's start with missense first, the middle one here. So a missense mutation would be if I changed one nucleotide and put in a different nucleotide, and now I coded for a different amino acid. So maybe I was originally supposed to have proline, and I've changed one nucleotide, and now I have serine instead. A nonsense mutation would be where I was supposed to have an amino acid, but now my mutation has introduced a premature stop codon. I have a stop codon earlier than I'm supposed to in my protein. So instead of changing from one amino acid to the next, to a different one, like in a missense mutation, now I'm changing an amino acid to a stop codon in a nonsense mutation. And then finally, a silent mutation is a harmless mutation. It's where I'm changing one nucleotide, but I'm still coding for the same amino acid. So remember from the codon chart we looked at a few days ago, multiple codons code for the same amino acid. So sometimes you get lucky and you have a mutation, but you still code for the exact same amino acid. So we can look at a couple more examples of this. So up here is my normal non-mutated situation where I have my RNA and then the appropriate amino acids that are made from it. So if I look at this first example, this would be an example of a silent mutation, because if I change this C right here to a U, both GGC and GGU both code for glycine. So that mutation had no effect, so that's a silent mutation. Here, I'm changing this G right here to an A. So instead of getting glycine, I'm getting serine. So that would be a missense mutation. I'm getting a different amino acid. And then finally, if I change this A to a U, I'm introducing a premature stop. So that would be a nonsense mutation. So we could also look at different types of frame shifts. So if in this example right here, I have the same original RNA, but now I'm cutting out this U. So now, instead of reading these three, and then these 3, now I'm going to read the same first 2 amino acids, the same first two codons, but now instead of reading UUU, I'm reading UUG. And then instead of GGC, I'm reading GCU. So this frameshift has affected every amino acid after the mutation. Similarly, if we look down at this bottom one, the bottom example here, I'm doing a deletion or an insertion of 3 nucleotides instead of just 1. So again, this one's less harmful because I am not shifting my reading frame. Just like up here where I read UUU and then GGC, I'm still reading UUU and GGC. I've just skipped one whole codon, one whole amino acid. Okay, so those are all mutations looking at kind of one base or a couple bases at a time. Other types of mutations you can have are much bigger scale mutations that affect an entire chromosome or an entire part of a chromosome. So the types that we're going to look at are a deletion, where I delete an entire chunk of a chromosome, a duplication, where you duplicate an entire chunk of a chromosome, inversion, where you take a chunk of a chromosome out and you kind of flip it over, you put it in the reverse order. And then there is also a fourth type called translocation that we'll see a little bit later on in this presentation. So let's look at some pictures of these. So here I have a chromosome. Oh, I don't mean to move that. I have a chromosome, and remember this chromosome is thousands and thousands and thousands of nucleotides long. So in this case, here's my original chromosome, and my mutation is that I'm cutting out an entire chunk that's probably hundreds of nucleotides long. So that would be a deletion. A duplication is where I take a section of that chromosome and accidentally either copy it twice, so I have a duplicated section, or sometimes a piece of a chromosome is removed, maybe due to a crossing over, and accidentally stuck on its homologous pair. So now I have 2 sections of the same chromosome on one chromosome. And then finally, the last type is an inversion, where I take this little piece here and remove it, maybe again to do a crossover, and then accidentally flip it over. So you can see where I have the red section first and then the green. Well, now I have green and then red. So these are mutations at a much larger scale. And the final type we're going to look at is called a translocation. So this is where you basically remove a section of a chromosome and then accidentally stick it onto a different non-homologous chromosome. So maybe, I have chromosome 1 here, and then I have chromosome 3. So these are not homologous pairs. And, I'm undergoing meiosis, and I'm trying to cross over my 2 chromosome 1, so I have a little break, and those pieces go to switch spots. But this piece accidentally gets stuck onto the wrong chromosome. It gets stuck onto chromosome 3, for example. So that would be what we call a translocation. So you could see here, this is an example of what's called a reciprocal translocation. So that means I break off the piece of 2 non-homologous chromosomes, and they both switch pieces. So that means the reciprocal means they're both doing it. They're both obtaining a piece from the wrong chromosome. Okay, so here I want you to pause your video and try to identify what types of mutations are in these 4 pictures. So go ahead and pause. Okay, so now let's go over the answers. So in this one, you could see that my original chromosome, it looks like 6 genes, the A gene, B gene, C gene, all the way through I. And then you can see that I'm deleting this middle section, so this would be a deletion. In example B here, I'm taking this middle section and I'm flipping it upside down. So instead of going A, B, C, D, E, F, G, H, I, now I go A, B, C, F, E, D, G, H, I. So this would be an inversion. So make sure you're checking your answers. In part C here, I'm taking a piece of this chromosome and I'm adding it on to this very different chromosome. So this would be a translocation. And in this one, I'm repeating this FGHI section, so that would be a duplication. So finally, one last question to end this video might be, okay, if I have a mutation in a gene, and that gene's important, will I die? Will it affect me to the point that I can no longer live? And this is kind of like a maybe for your answer. So one, it depends on how bad the mutation is. And secondly, remember that we actually have 2 copies of every gene. Remember, we're diploid organisms. So if you have 1 normal copy and 1 mutated copy, you're okay if that mutation is recessive because you still have the dominant normal copy. However, sometimes mutations can be damaging enough that they can be they can actually be a dominant mutation, and that's a more severe situation. And it would be harder for an individual to survive with a dominant mutation in a very important gene. So that's video 8. See you in video 9.